Alrighty, everyone, what we're looking at is the schedule for this semester. As you can see, it's generally been what we've been talking about. We're slightly or going a little bit slower than what we usually would be, but we're going to be able to catch up pretty easily. So this is where we're at right now, which is what we're going to be starting, but we also do need to still finish off the cell. So we're going to talk about mitosis and meiosis, the cell cycle, but then finish up the cell as well. But an important date that you can see is coming up is exam one. So exam one is going to be on February 19th. That should be 2 weeks from today. So it doesn't, or it kind of seems like it's far away, but this would be the time that you want to start preparing. Like, if you haven't been looking over the materials so far, please try to do so because that can creep up on you, and this can be a little dense if you're not used to studying for science classes. So the homework, once again, should help with making sure that you have some idea of what we've been talking about with intro to biochemistry in the cell, but you will have additional homework that'll be based off of many of these materials as well. So this should be relatively aligned with lab, not completely, but we're doing our best. Like, that's just a little difficult. But at the very least, you can see that our exam is coming up. So just make sure that you're preparing appropriately. So, well, I guess, does anyone have any questions about anything? Oh, good question. So that is actually something that I wanted to gauge the class on, but I guess I'll do that on Wednesday because we could do it in one of two ways. If we can, like, we could do them on Canvas, but in person, as in like still using Proctorio, just not or not Proctorio, Lockdown browser, just not with your camera. Because that would be, I mean, it's very easy to get your scores afterwards. It's very quick, it's very simple. But we could also do it on Scantron as well. So I'll see how the class feels about it on Wednesday, and at that point, we'll figure it out. But either way, the exam is going to be mostly multiple choice with some amount of short answer. So, but remember, short answer is like short, like very short, as in, you know, short sentences, fill in the blank. That's that's it. No paragraphs, no pages, nothing. So short. Any other questions about anything? So there's a question regarding whether you need to turn in the PDF, and the answer to that is no. So, I mean, doing the PDF, printing it out and filling it out is really helpful. That's what it's meant for. You can do it, well, on paper, or you can do it on like a tablet or otherwise. But if you do that, that should help with preparing you for that quiz. So you don't need to turn in the PDF. You just need to turn in or take the quiz. Any other questions? Alrighty. Well, then I guess with that said, let's go ahead and start today's material, which, if I recall correctly, we were still talking about the cell. So what we can see here is the cell. And we talked about the plasma membrane. We talked about the barrier. We talked about its variations, as in microvillus, cilia, as well as the flagellum. But now we'll talk about, well, our next major part of the cell, which is called the genetic material. So the genetic material, that's going to include the, hmm, one second. There we go. So the genetic material, that's going to include basically the nucleus and then everything that it needs to pass on to the next cell in order for it to be able to function properly. So the way that cells get the basically blueprints, as in how to be a cell, what to do as a cell, like that's going to be the nucleus and the genetic material. So to take a look at our genetic material, we start off by taking a look at the nucleus. And this is a very large part of many cells. As you can see, it forms this really big blobby thing right near the center of the cell. This right here would be the nucleus. The nucleus is going to hold the genetic material. And basically, this is kind of like the command center. This is where everything, all of the directions come from. So you can kind of think of it like the brain. So everything that the brain or that the cell is going to do, many of its functions are going to start here from the DNA, and then it's going to send out directions using something called RNA to tell the cell what to do. Now, a key thing about looking at the nucleus, though, is that there are multiple parts to it. So the whole thing is the nucleus, but inside of the nucleus, this is where we have the DNA. And the DNA comes usually in a form called chromatin. So chromatin is a form of the DNA. So if you look closely, this right here is the DNA. You can see it in its typical, double helix form, the way that you usually see it. But this DNA is all throughout the inside of the nucleus. So all of this stuff that looks like a big bundled up, tangled ball of yarn, that's actually the DNA. And the form that it's in right now is called chromatin. So chromatin, just to kind of bring up something, that actually might sound a little familiar to you, as in like, well, not familiar per se, but something might sound similar to it, which is chromosomes. So if you look closely, chromosome right here, do you see this X-shaped structure? This is probably what you've seen genetic material as, like whenever you see it, besides like the double helix. But just be clear, these chromosomes are made up of DNA, and specifically chromosomes are just very, very tightly wound DNA. As in like this DNA is like very, very tightly packed into these little condensed forms called chromosomes. But when the DNA is not compact, that's chromatin. And that's usually the case because chromatin, as you can see, is very spread out. This is going to allow the DNA to actually do stuff. Like it's not going to be really tightly packed because if you tightly pack something, you can't really access it very much. So chromatin is the one that you can see here. This is going to be the form of genetic material, which is generally throughout most of the cell's life, except for cellular division. Oh, okay. So moving on to our last part then, we have cytoplasm. I feel like, did we miss anything? Did I skip something? No, I guess not. Okay, good question. So the question is the chromatin inside the nucleolus or what surrounds it? And to answer that, the nucleolus is kind of like a large aggregation of the chromatin. So going back to the previous slide really quickly, this right here is called the nucleolus. And you can see the chromatin coming off of the nucleolus. This is just kind of like a bunch of the chromatin all together. And the nucleolus is usually a visible structure which looks like a little ball inside of the nucleus, or the nucleolus is a little ball inside of the nucleus. So the nucleolus is technically made of chromatin, but it's just like a bunch of the chromatin all condensed together, but not as dense as chromosomes. So that is a good question. And I know like these terms do kind of like blend together a bit, but at the very least, I would say that the nucleolus and the chromatin can almost be said to have like the same function because they are, I mean, basically the same thing, just the nucleolus is what you actually visibly see as a part of the chromatin. Like when you look at this outer part, when you look at a cell, you don't usually see this stuff, but you can see the the nucleolus, which is made up of chromatin. So good question. And if it comes to it, I'll just make sure that I ask it very clearly if that is going to be something on an exam. Any other questions? Well, just put them in the chat if you have questions, but let's keep on going. So our last part and the majority of the cell actually is called cytoplasm. And just try and think back, dig back to Wednesday. What does cyto mean? Cyto means cell. So, like whenever you see cyto, that would be a good idea to think of cell. Oh, shoot, micro. We're not using micro yet, but cyto would mean cell. And there's three terms, I believe, at least, that we're going to be seeing regarding inside the cell: cytoplasm, cytosol, and then another one called cytoskeleton. But we'll get to that. So cytoplasm, if you look closely, is everything except for the nucleus and the plasma membrane. So, I mean, the plasma membrane we talked about, the nucleus we talked about, everything else, all of this other stuff, that's called cytoplasm. So even though there's like a lot of variable stuff, lots of wiggly, or not wiggly, but curved things, round things, crunchy looking things, like all this stuff except for the nucleus, this is cytoplasm. So cytoplasm includes this stuff, or this fluid-like stuff called cytosol, and that's this gel-like substance, but then also all of the organelles in the cell as well. So kind of think of like how we have organs. Think about how we have a stomach. Think about how we have a heart, intestines, or blood vessels, muscles. Those are all part of the cytoplasm, except for the nucleus. So, I mean, the cell has its own little things called, instead of organs, organelles, which are basically like its stomach, its transport system. So that's going to be all these little things except for the nucleus. But just to once again bring this up, cytosol is a little different from cytoplasm because cytosol is a little more specific. It's only this gel-like substance or fluid inside of the cell. So basically, cytosol plus all of the organelles, that's cytoplasm. So once again, organelles, they're basically little organs. So just like how we have organs that are doing various major functions for us, the cell does too. The cell does the same thing. Like it breaks stuff up into making proteins, making fats, making energy, packaging this stuff and sending it throughout the cell. So all of the little like organs that we have, generally there's some sort of organelle that does something like that for the cells. So whenever you see like the word gets longer for some reason, usually it's smaller. So we have organs, cells have organelles. So to start off with the doozy, at least audibly, we have the rough endoplasmic reticulum. And you might see this abbreviated as RER sometimes, but it would be good to know the words. So rough endoplasmic reticulum, this is basically this thing that is on the outside of the nucleus. And this layered structure with all these little bumps, these rough surfaces, that's the rough endoplasmic reticulum. Now, the key thing about the rough endoplasmic reticulum is twofold. Number one, how do you identify it? Like, that's a good question that you want to ask yourself, especially in lab. How do you identify it? Because a lot of these things, when you look inside of a cell, I mean, you can't really recognize them very clearly unless you know what you're looking for. So some stuff to consider. What's it next to? What are some key characteristics that help you to identify it? And then also maybe a little bit of what does it do? So, looking closely at the nucleus, this structure is actually connected to it. Do you see this little connector right there and right there? This is directly connected to the nucleus and it's directly outside of it. So if you look for that, I mean, that would be definitely a good note that I would take. But even more so, what does its name indicate? Rough endoplasmic reticulum. That means that it's rough. That means that it has a like bumpy surface if you look closely. And the reason for that is because it has these little they're little machines called ribosomes that basically make proteins. But in the rough endoplasmic reticulum, when those little things make proteins, the rough endoplasmic reticulum can modify those proteins. It basically kind of like helps to cut it up, readjust it a little bit into the more stable and mature state. Now, if you have a rough endoplasmic reticulum, you also have smooth endoplasmic reticulum. But once again, questions that we want to ask, how do we identify it? What's close to, or like what's close to it? What does it do? So if you look closely, how would you describe this one? So the rough endoplasmic reticulum was right next to the nucleus, but the smooth endoplasmic reticulum is a little bit further away. So it's kind of like the second layer of these like, well, blobby layers, I suppose. This is the smooth endoplasmic reticulum. Now, furthermore, the smooth endoplasmic reticulum, what does that look like? What does its surface look like? It's smooth. So no bumps, no ribosomes. And if you remember, ribosomes make proteins. That's not what our smooth endoplasmic reticulum is doing. Smooth endoplasmic reticulum is actually going to be involved with two major things: making lipids, as like making fats, kind of like steroids and steroid hormones, but also detoxifying stuff in the cell. So that would could include stuff like, well, specifically poisons, as in literally alcohol is a weak poison to us. But yeah, alcohol, various drugs, the smooth endoplasmic reticulum can help with, well, detoxifying it to some extent. So remember, big difference between them though. Rough endoplasmic reticulum, bumpy surface; smooth endoplasmic reticulum, smooth. But then furthermore, rough endoplasmic reticulum processes proteins; smooth endoplasmic reticulum, lipids and detoxification. So try to make those kind of like distinctions between them so that you can have it much more clear in your mind. Now, to talk about those ribosomes, those little bumps on the RER, these are the things that are making proteins. And if you look closely, this little blobby structure, this is actually one of these tiny little yellow pearls. So this ribosome is actually going to be making proteins. And if you look closely, to just very, very briefly describe how this works, do you see this thing called mRNA? This is actually kind of like a transcript which came from the DNA. It's not the DNA, but it's kind of like a modified copy of it. And this mRNA, like when it reads the mRNA, it's literally telling it how to build a protein. It's telling it to build or like add this protein or not protein, add amino acid type A, B, A, B, C, A, B, and so on forever. So that's a very simplified version of this, but you're literally just chaining amino acids together, which then eventually make our polymer, or yeah, polymer, which is going to be the protein, the peptide, or yeah, the big proteins. So these ribosomes you can see all over the place, though. They're on the rough endoplasmic reticulum, but just to be clear, there are also these things called free ribosomes, which are just floating around in the cytosol as well. So just to be clear, ribosomes are the ones that make the proteins. The rough endoplasmic reticulum modifies those proteins. So once again, make that distinction because they are different. Now, a fun one to say as well, the Golgi apparatus. The Golgi apparatus, if you look closely, is kind of blobby as well, but once again, how do you distinguish it? So, number one, it's a little further away from the nucleus. It's not like RER or SER, but for its shape and structure, it's basically like this stack of pancakes. Like, I mean, you can think of it however you like, but you can see that it's just like these stacks of thin shaped folds, and this is just a stack of pancakes. So the stack of pancakes here, this is basically kind of like the packaging center of the ship or of the cell. This is how it ships stuff out, like away or to different parts of the cell or even out of the cell. But in order for this packaging center to do this, it also typically has these little boxes, or rather bubbles, that it uses to send them out. So first of all, once again, this is the Golgi apparatus. But then what are these things coming off the Golgi apparatus? Those are called vesicles. So vesicles, those are the little, basically kind of like the boxes that this packaging center uses, but they are actually bubbles. And just to also be clear, these bubbles are actually made up of something very similar to the plasma membrane. It's much simpler, but it's literally a little kind of plasma membrane that's surrounding whatever you're trying to send out. Maybe you're trying to send out a fat or a protein or both, and maybe you're trying to get it to a certain part of the cell. Those vesicles will float through the cell in order for it to reach that part. Or potentially, you could even go to the outside of the cell, fuse with the outside of the cell, and release it. And if you remember what that's called, that would be exocytosis. So when, once again, we're identifying these things, though, try to kind of group them together. So whenever you see a Golgi apparatus, what are those things that are right on or right outside of it? Those would be vesicles. But vice versa, if you see those vesicles and they're all aggregated around something, that's probably the Golgi apparatus. So if you look closely, I'm trying to connect these things so that I can find them more easily and I can be a lot more confident in what they are. Not that I'm going to ask you that in lecture, but it also helps to remember their functions too, I suppose. So that's going to be how you can group them together and figure stuff out. Now, one that many people have actually heard of, the mitochondrion, but the word that you probably use is mitochondria. So just to kind of give you a distinguishing, or just to distinguish those two terms, the mitochondria is actually the plural form. So mitochondrion is singular, mitochondria is plural, but take a look at this organelle again. So just to be clear, then you probably have heard that this is the powerhouse of the cell. And what does the powerhouse of the cell do? It makes ATP. And we know what ATP is. This is the cellular fuel. This is the cellular energy that you use to basically do cellular activity. So this is going to convert like sugars and then oxygen to make that ATP. But just to also be clear, as a byproduct, it also makes carbon dioxide. So the carbon dioxide that arises in our body, a lot of that is coming from our cellular respiration or our process of making ATP. But once again, how do we identify this? I mean, we know it makes ATP, but you don't typically see little ATPs coming out of it. So what's different about this? And if you look closely, it's kind of like a bean shape, not the most helpful, but what does it have inside? It actually has this folded inner membrane. This is called cristae. And that's not an important term to know, but knowing that it has that double membrane, knowing that it has this folded inner lining inside of it, that's going to be making it easier to identify. So if you see a bean shape with this stuff inside, I mean, that's a good indication of a mitochondrion. All right, now the one that's honestly the most difficult to identify, just because it's the most generic, it's the lysosome. So you can see closely that these are just like these scattered medium-sized balls. And unfortunately, that's all I've really got for you. So you really do have to use a little bit of elimination, just a slight a bit of strategy in identifying it. But at least its function is very distinct. Its function is actually to break things down using enzymes, if you remember those. Enzymes, those things that help with pushing forward chemical reactions, those reactions in this case are breaking stuff down into smaller components. So breaking stuff down inside of the cell, that is going to be something called autophagy. So autophagy, when you digest and remove materials from the cell, I mean, that's kind of like our stomach, as in like we have a stomach, or rather our digestive system, but our stomach is the one that we know it. It breaks stuff down. It uses chemicals to break them down. It uses enzymes and acids. And eventually, a lot of that stuff gets really small and we either use it or expel it from the body. But there is one more thing that kind of comes up here is something called autolysis. So do you remember the word lyse? We only used it very briefly when we talked about cells exploding. So lysis is the process of a cell exploding. And if it's time for like your cells to undergo this something called like pre-programmed death, as in like they get too old, so they actually like kind of ultimately kill themselves to some extent, they're going to release their enzymes from their lysosome, and then that's going to digest the cell to ultimately kill it. But once again, it's breaking things down, and then ultimately, at least hopefully, you'll be able to use those contents for other cells instead. So at least identification wise, a little bit generic, but otherwise, very important and very interesting organelle. Now remember the word cyto again? Cyto, that means cell. And guess what cytoskeleton is? If you know what the word cyto means, it's the cell skeleton. So in this case, you can see all of these tiny little tubes all throughout the cell. That's what it's made up of, is these things called microtubules. And that's going to allow it to basically give the cell certain shapes. Like it's going to give certain shapes. It's also going to help with moving stuff in the cell, especially during cellular division. So cytoskeleton is exactly what it sounds like. It's the skeleton of the cell. So shape, support, and movement in this case. And then something similar, but a lot more specific, we have this thing called centrioles. So look closely. Do you see this pair of structures here? These are centrioles. And if you look closely, this process right here, where the centrioles are actually being used, where they're actively doing something, this is mitosis or cellular division. So centrioles are something that's going to aid specifically in cellular division. But once again, how do we identify these? And luckily, they are pretty distinct. But if there's something that can help you remember it, like try, I mean, use it, think of those things. And sometimes they could be silly because centrioles, at least for me, look like the mini churros. Now, obviously, don't write mini churro on, an exam or a practicum, but if it helps you to identify it, if it helps you to get your mind started a little bit, those things really can count. So do what you need to, but yeah, the centrioles, they are the little mini churros of the cell, and those are active during cellular division. Now, to finish up the cell, we have a few more processes just to talk about really quickly. So we actually talked about that already. Oh, we talked about all of this already. So hold on. I think it might be in the wrong PowerPoint. But we do have one last thing to talk about, which is cellular respiration. All right, so we already talked about all the cellular movement stuff. We talked about the channels, the osmosis, the tonicity. We talked about that during chemistry. So let's go ahead and now talk about something that's very important to the cell, cellular respiration. Now, once again, a lot of these cellular things, they probably don't sound very important or maybe at least familiar, but at the very least, just think about the word respiration in your mind. Like maybe think about what comes to mind regarding respiration at all. And the one thing that can probably come to mind is gases, as in like maybe oxygen or yeah, oxygen and breathing. And that's actually really important because it is related to this process, cellular respiration. So cellular respiration, just to be clear, this is the process in which we make ATP, our cellular energy. It's really important. Like without ATP, your cells aren't going to be able to do most of the functions that it needs to. So this important process is crucial to everything, basically. Now, just to give you some very general breakdown of what cellular respiration is going to include, you can see that it has four major phases. Now, there's much more to this, and I will never really ask too much about this, but these are phases that I would like you to at least be familiar with. Maybe the order of which or the names, because this is how your cellular respiration is going to occur. So, do you see the word lysis in here again? Glycolysis or glycolysis. This is when you break down sugar. So, when you eat foods, you're using it for energy. This is where it happens. That glucose is being broken down into different forms, and that is going to be using oxygen along the way. And then eventually, the things that you make here are carbon dioxide and ATP. So, actually, this is an important part as well. So this little equation here, where you have one side and then an arrow going to the other, this is cellular respiration, basically. This is the basic units, and then this is what you use, and this is what you make. So I would definitely try to be aware of at least the stuff that you use and make. So glucose, this is where we use our sugars. This is where we use or break down our foods to get our energy. So glucose is being broken down. But remember, cellular respiration, we also need to use oxygen. So, very important to breathe, right? If you don't breathe, you're in kind of big trouble. So, you need your glucose, you need your oxygen, and then you use these two through cellular respiration to make ATP, the important part, but also a waste product, carbon dioxide. So, when you say like you breathe out carbon dioxide, that's this right here. When you make it in your body to make ATP, you have to get rid of it. And the way that you do that is by putting it in the blood, bringing it to your lungs, and then breathing it out. So don't worry, there's a lot more to that that we'll focus on a little later in the respiratory system, but glucose and oxygen being used, carbon dioxide and ATP being made. And then these four phases of cellular respiration is basically the very bare bones that I would like you to at least be familiar with. So to break this down a little bit, no, once again, not stuff that I would want you to focus on, but glycolysis. This is where you're breaking down the sugar. You break down the glucose and then you make these things called pyruvate, and then that is going to help with forming some ATP. So this is how you make a little bit of ATP, but that's not much. 2 ATP is good, but not what you want necessarily. So what you can actually do is then use these two pyruvate molecules in something called the transition reaction. So this is where you'll make another form of pyruvate called acetyl-CoA. And acetyl-CoA, once again, is not something that I necessarily need you to know, but this is important because this is going to be used in our third phase called the citric acid cycle. Now, this is where we're going to make a little bit more ATP. We're also going to make a little bit of carbon dioxide. But then lastly, if you look closely, there's all these little things that are along the way that I'm not going to talk about, but NAD, NADH, FAD, FADH, this stuff is going to be really important for our last part, which is actually the most important part, called the electron transport chain. But to be honest, if I were to be a little bit specific in things that I would want you to know, the terms glycolysis and electron transport chain, those are really important parts. So electron transport chain, once again, I'm not going to explain this in all of the chemistry and molecular or biochemistry aspects of it, but the main fact of this is that electron transport chain, this is the part that needs oxygen. So you need oxygen at this point, but this is also when you make most of the ATP. So far we've made 2 ATP here, 2 ATP there, but the electron transport chain is where you make 32 ATP. So this is how you make most of your energy. Remember, glycolysis makes pyruvate ATP. Transition reaction makes acetyl-CoA and NADH. You make a little bit more ATP in the citric acid cycle, but then all these NADH, FADH, and so on, that's going to be used in the electron transport chain, and that's where you make ATP, or most of the ATP, 32 ATP. So remember, we have, believe it or not, very fast metabolism. As humans, we have to generate a lot of energy, we have to generate a lot of heat, and to do that, we generally need a lot of nutrition, like sugar, but then we also need a lot of oxygen. We definitely aren't lizards. We can't just lie down and not use a lot of energy. We are endothermic. We need to make our own energy, our own heat. So yeah, cellular respiration, big thing. So this is going to be something that comes back, at least the whole process, not necessarily all these little parts, but know that cellular respiration uses sugar and oxygen to make carbon dioxide and ATP. And at least being a little familiar with these names, as well as the major things that happen, like making a lot of ATP, using your sugar, those would be what I would focus on. You can kind of see this process here. Glycolysis happens in the cytoplasm, and then it moves into the mitochondrion, where everything else basically happens, so that you can then make all those ATP. Now, lastly, what happens if you don't have oxygen? So you can still break down glucose without oxygen, but this process is known as fermentation. And if you've ever felt like a kind of burning in your muscles when you don't have enough oxygen, or when you overuse your ATP and you cannot make enough of it fast enough, you probably have heard of and maybe felt this phenomenon where you make an acid inside your muscles called lactic acid. So this is not a very efficient way to make ATP though. Using fermentation, you only make 2 ATP at a time. Remember, you made 36 ATP in total from cellular respiration. But from fermentation, every cycle or every process only makes 2. So you're still making ATP. I mean, you definitely need it if, or need to do this if you have no other choice. But if you have oxygen, if you have sufficient oxygen in that tissue, you can do cellular respiration to make ATP a lot more effectively. And then when you have like no oxygen and you undergo fermentation, remember, there's another byproduct that can start to build up, lactic acid. So with that said, I think that should be just about it. Oh, shoot. Also, I'm going to be posting the study guide very shortly, so please make sure that you take a look at them. But I mean, this is just kind of like a sample of what you can expect. So it's not going to be like question by question, like stuff that you can, obviously it's not going to be questions of the exam, but it is going to be stuff that helps you to organize the material to see if you're looking at everything at a level that is going to be sufficient to do well on the exam as well. So with that said, that should be it for the cell. Does anyone have any questions about anything before we move on to our next lecture? Alrighty. Well, if you do, once again, just throw it in the chat. But let's go ahead and talk about what these cells can do throughout their life and how we make more cells specifically. So what we're going to be looking at is the process of the cell cycle, the usage of cell division, and then how chromosomes are going to be passed on from cell to cell and in different types of cells as well. So let's go ahead and get started. So just to get started, just to be clear, we begin life as a single cell. Like when a sperm and an egg combine together, that makes something called a zygote, and that's one cell. I mean, those two things combine to one cell, and that's where we all start. But obviously, we have to make more and more and more cells to eventually get to the trillions of cells that make the adult you. So the way that we're going to do this is through two different cellular divisions. So the main one is called mitosis. That's the one where we make more generic cells, like cells that you use throughout the body. It could be like a skin cell, a muscle cell, or otherwise. But we also have a little bit of a special type of division called meiosis. And that's the one that makes the gametes. And that's specifically for making sperm and eggs, basically. So they are going to be a little bit different in how they divide to do that. So, I mean, we'll talk about that, but let's go ahead and keep going. Let me get laser pointer. So let's talk about mitosis. So mitosis, this is the process of cellular division. And just to be clear, when things divide, things usually get smaller and smaller and smaller, but through the cell cycle, like it actually replenishes itself along the way. So what you can do though is like take one cell and then divides into two daughter cells. And then a key part about mitosis and this division specifically is that you should be making a very similar cell to the original. As in like the cell that is going to be made from this process should be almost the same. As in like it should have the same number of chromosomes, it should have the same types of organelles, and it probably will be doing a very similar function to its predecessor. So what we call these are identical daughter cells. So these cells are considered the same, basically. This is how we make more cells. And this is important because, number one, we need to grow, but also when we lose cells, we need to repair as well. As in, like, when you scratch your skin, like, you do lose cells basically every time. So to prevent you from, scratching off all your skin, Your skin needs to be able to make more cells and repair itself. And then meiosis, on the other hand, like I was saying, this is going to be making a certain type of cell called a gamete. And these are the oocytes, or the eggs, and the sperm. Now, a key thing about sperm and oocytes, or sperm and eggs, though, is that they only actually have half of the genetic material, or half of the chromosomes of a typical cell. And why would that be? Like, why would you only want half of the genetic material? It's because when you have a sperm and an egg combined together, one half and one half makes one whole. And that's going to be allowing you to make the combination of genetic material between mother and father to make, well, you or the babies. So, to answer the question, are the daughter cells the same as each other or identical to the parent cell? Kind of all of the above. As in, like, the parent cell is actually one of the daughter cells still, because when you have one cell, like, it's going to divide into two, and then one cell is going to be kind of like the original, and then one cell is going to get a copy of everything. So the daughter cells are the same as each other, which should be the same as the parent cell as well. And this can get a little confusing because, lots of cell stuff, lots of small stuff. So if you have questions, let me know. And then, unfortunately, some of these terms can be a little bit overlapping. So if you have questions about that too, let me know. So to get to some of that stuff, let's talk about the genetic material. So we talked about genetic material, we talked about the nucleic acids, and we talked about how it makes DNA. So deoxyribonucleic acid, that's DNA. And DNA, if you look closely, is that double helix form made up of those nucleic acids chained together. But keep in mind, like, this DNA does not look like, or you don't see it like this typically in the cell. You either see it as chromatin, or as you can see here, chromosomes. But the key thing about, like, what's on the DNA, what the DNA like literally is, it's made up of these things called genes. So genes. A gene is basically a unit of DNA. It's a unit of heredity, and could be a very long unit of nucleic acids that are going to, and okay, just backing up. This unit is going to provide a function for the cell. As in, like, this string of nucleic acids might tell you something specifically, maybe like, to be very simple, what hair color you're going to have, what eye color you're going to have, or maybe how fast you grow your nails. It could be like these very minute things, but you have genes for generally everything to make sure that you can tell the cells and tell the body what to do. So everything that makes you, well, most of what makes you is due to the genes that you have in your DNA. So those genes are going to be like the map that you use to, or the script rather, that you use to tell the cells what to do to make you. Like, let's make their hair this color. Let's make this amount of pigment. Let's make a lot of muscles, or, make these hormones, or even be as small as, like, make this type of transporter, or make this type of cellular receptor, or something like that. So these genes can be really important or really minute, and they are what's making up the DNA. Now, to once again bring this up, DNA has multiple forms. As in, like, depending on what the cell is doing, you might see the DNA in a slightly different shape. So the one that we talked about previously was chromatin. So looking at this cell, do you see this big bundled up yarn that's just everywhere and disorganized? That's chromatin. But the reason why it's unraveled and really all over the place, because that's how you're going to allow it to be able to be accessible. That's going to allow it to be active. So this part or this form is chromatin. This is what's going to allow the DNA to actually do stuff, basically. So this is going to be the part that you actually can use. But if you're trying to condense everything, if you're trying to move it, like, just to be clear, like, this might be like in your house, you unpack everything because you want to use it. But if you want to move, if you want to like keep everything organized or not lose anything, you're going to like pack it all together and condense it. Like, kind of think of it like putting it into like boxes. You're going to organize everything, you're going to condense everything into very specific spots, and that's how you get the chromosomes, basically. So when you wind up this chromatin very, very tightly, you're eventually going to form these little X-shaped structures, which are called chromosomes. So when you want to move genetic material, if you want to have specifically cellular division, you want it to be in the form of chromosomes. So hopefully that made sense. If you have questions, once again, let me know. But this is where it can get a little bit tricky. So let's talk about types of cells and names for names for chromosomes. So just to kind of give you a intro, when you say the word soma in somatic, we're talking about the body, and this is the most generic type of cell. So these are basically everything except for the eggs and the sperm. So everything except for the eggs and the sperm, whether they are bone cells, muscle cells, blood cells, skin cells, like generally, they are all somatic cells. And these somatic cells, like you want them to have all the genetic material. You want them to have the whole script so that they know what to do, where to read from, and do what it needs to do. So these somatic cells have all of the chromosomes. Like, yeah, all of them. And just to be clear, humans have a total of 46 chromosomes. Or just to kind of give you it in a uh different perspective, 23 pairs. Now, this is a number that I want you to kind of be aware of. Like, if you have a highlighter, if you want to circle it, this would be a good place to do that because this number is going to come back a couple times. So 46 chromosomes total, or 23 pairs. So when I say pairs, I mean like two. So 23 times two, that's 46. So if you want to use a calculator, go ahead, but I promise 23 times two, 46. But the reason why I'm talking about them in pairs is because they usually come in something called homologous pairs. So we have 46 total chromosomes, but we actually have 23 types. So 23 types of chromosomes. So that means that we have like chromosome type 1, 2, 3, 4, 5, and so on. You can see them kind of like listed here. But then we have two of each. Why do we have two of each? Like, if you look closely, one of these came from your mom, one of these came from your dad. Like, remember, half of your genetic material comes from each parent. You have one type 1 from your mom, one type 2 from your dad, one type 2 from your mom, one type 2 from your dad, and so on. So these are how you get these things called homologous pairs, which are pairs of our 23 types of chromosomes. But remember, 46 total. So try to keep track of that number, but also let's keep going. So These are 23 types of chromosomes which are paired together because you have one from each parent, basically. We have a term for this, though, where we have two chromosomes of each type. This is called diploid. So for the most part, we are diploid. Like not everything is diploid, but we are for sure. So what that means is we generally have two of each type of gene, as in like you have one form of this gene from your mom, and you have one form of this gene from your dad. And that can form like very different interactions, which we'll talk about in genetics. But you can see that like for each pair of chromosomes, you have these different genes from each parent as well. So that means that for every gene, for every chromosome, we generally have two. Now, this can come with like one exception. So hold on, before we get to that, we have two major types of chromosomes. Now, the ones that I've been talking about basically are called autosomes. So autosomes, those are the ones that are generally the generic one. Like obviously there's different types of each of them, but for type 1 through 22, those are all autosomes. Those are all going to give you different genes, different characteristics, depending on where it is, what chromosome, but those are all autosomes. Those should be pretty similar to each other. Those are specifically not sex chromosomes. So sex chromosomes, those are a little different. Sex chromosomes, there are two major types. Like, and those types are pretty different shape and size wise. So you may have heard of these before, like X chromosome versus Y chromosome. Like the combination of those two is going to give you, well, genetically, are you a genetic male or genetic female? So genetic as in like what chromosomes you have. So just putting a label on it like genetic female or genetic male. So what you inherited from your parents in your cells. So XX, this would be genetic female, XY. This would be genetic male. And hold on, do I have a picture of it? No. Let me just go back real quick to show you how different these are. You see these two chromosomes here? This is our 23rd pair, our one pair of sex chromosomes, and the X chromosome is much larger than the Y chromosome. So just to be clear, like genetic male would be XY, so this person would be a genetic male. But if it had two big X chromosomes, it would be a genetic female instead. So most of our chromosomes are autosomes, but we have one type of special chromosome, which is sex chromosomes. And that's the one that gives you your genetic sex. Now, to start talking about the process of cellular division, we have to start by talking about the cell cycle. So, the cell cycle, this is kind of like what the cell does, well, throughout its normal whole life, basically. Like the cell goes through this cycle repeatedly until it eventually dies through from old age or otherwise. But if you look closely, it has these large phases of life where it's just doing its thing, like doing whatever it's supposed to be doing, whatever, whether it's a muscle cell, a bone cell, or otherwise. But then there is the potential mitotic phase, which once again is cellular division. So this is, normal life, just doing its thing. But then if you need to make more cells, which typically you do, you have the mitotic phase here. So let's talk about the cell cycle a bit. So if you look closely, there are 3 or 4 major phases. G1, okay, let's have a starting point too. So let's just say that you had cellular division just occur. And let's just say that this is like a fresh cell. This cell is ready to do its thing. It's going to start off by going through G1, S phase, G2, and then potentially another round of mitosis. So if you look closely, there are some things associated with each phase, though. G1, luckily, like the G in G1 is growth. So growth. But this growth is just like, as normal things would. Getting bigger, doing its thing, like finding its stride. But then something that's really important, especially to prepare for mitosis, is synthesis or DNA synthesis. So you had G1 growth, but then you have the S phase. S phase is the synthesis phase. And what's specifically happening here is you make a whole second copy of your DNA. Like you make a whole second copy of each chromosome. You make a whole second copy of each gene because before you get to cellular division, you want to have those two copies to give one to each of these two cells. Like if you only have one copy here and you try to split it between the two cells, only one or each cell is only getting like half of that map. Like that's not good. They are not going to be able to find their way. So you need to make another whole map. You need to make another whole script. And basically you synthesize a whole second copy of all of your DNA and genes. So G1 was grow, do its thing. S was make that second copy. You need to get ready. And then G2 is just more doing its thing and getting ready for cellular division. So another thing that happens in G2 though is getting bigger so that when you do divide, they're not going to be super tiny. Like if you just go from one normal cell and it divides into two small cells, those two small cells are not going to be super happy about that. But if you grow a lot so that you are a little bit bigger than normal, and then you divide those two cells that come off of that, should be in a little bit of a better place. So I know I just kind of like, I don't know, went over this quite extensively, but just to kind of like reiterate, G1, this is the growth phase. This is where it's doing its thing, getting ready. And once again, to keep track of this number, G1, this new cell has its whole set of 46 chromosomes. So this cell has 46 chromosomes during G1, just one whole copy of the DNA. Now, this is where it gets interesting because remember, you're making a whole second copy of the DNA. You're making a whole second copy of all the chromosomes. So how many did we have in a normal cell? 46. And then when you make a whole second copy of those 46, now this cell is actually holding on to 92 chromosomes. So you've got 92 chromosomes now. You have like two copies of the 46 total. So this is where we get a specific kind of like term that we use, which is sister chromatids. So this is where like the terminology kind of becomes a little bit overlapping. So just to be clear, these are all chromosomes. These are all chromosomes. But when you have copies of the chromosomes, we can call them chromatids. So chromosomes are, the chromosome, and then chromatids are more specific to being copies of the chromosome. And then when you have two copies of the chromosome, typically you'll see them linked together in the middle. So if you look closely, you have a chromosome A and a chromosome B. This is like when you have your 46. But let's just say you make a whole another copy of A and a whole another copy of B. Oh, wait, that's not a B, there's a lowercase a, but you get the point. But if you look closely, now you have 2 of the blue and 2 of the red. and they're linked together in the middle through using something called a centromere. And does this look familiar? Have you seen this before? This is what you usually see when you think of chromosomes. So this X shape is because you have two copies of this chromosome, or rather two sister chromatids, which are linked together in the middle and helps to form that X. So we'll come back to this, but let's just keep going and let's take a look at what happens next. So now you have 92 chromosomes in this cell after the S phase. And then now it's just going to keep doing its thing. The G2 is coming around and it's getting ready for mitosis and getting big, but still doing its thing. But remember, this G2 is still holding 92 chromosomes. Now, lastly, oh shoot. Okay, I think this is just showing like the formation of the chromosomes and the sister chromatids and so on. So once again, this X shape, this is what you kind of like expect to see with a chromosome. These are two sister chromatids, two identical copies of each other, and they're held together at this little thing in the middle called a centromere. So I know a lot more than what you thought with like chromosomes, but here, hopefully this will help as well. So this is a homologous pair. Oh man, I keep throwing these terms at you. So these are chromosomes of the same type, homologous chromosomes. That means that this could be maybe chromosome number 11, and you have one from your mom and one from your dad. You have to make a copy of both of these. So you have like a second version of this. And the way that you do this is just by replicating it during the S phase. But when you do that, you will basically have two copies. And those copies are chromatids. And when they're the same copy of each other, these are sister chromatids. And then they are held together in the middle at the centromere. Whew. So this is how you get from like one copy of every chromosome to two copies of every chromosome. This would be a cell with 46 chromosomes. This would be a cell with 92. Alrighty. So looking at this though, this is finally leading us to cellular division or mitosis. So mitosis, or rather, well, yeah, cellular division of somatic cells. This is how we make more cells. And to do that, we want to try and make, like, remember, identical cells, which just makes more of the same kind. So you undergo mitosis and something called cytokinesis, or cell movement, basically, where they kind of like completely break apart. But this is how we build the body, grow the body, and then hopefully maintain the body throughout your life. So this is where we're going to start talking about like the actual phases of mitosis, which comes in four major phases, but there are some major parts to this. So as you can see, we have our four major phases, which we'll talk about later. But just to kind of give you an option, would you like me to continue this and maybe have it in? No, okay, you leave right now. But do you want me to continue this and make like a longer video, or do you want to talk about this on Wednesday? I mean, otherwise you have to watch it later, but maybe I can go through it a little bit faster on Wednesday if I talk about it now. So if you have an opinion on it, go ahead and say so in the chat, but that's where we're going to stop for now. But otherwise, I could continue. That's all. Or if no one's there, that's fine too. But otherwise, okay, Wednesday sounds good. This is enough. All right. So we'll continue mitosis next time then. look forward to that. But thank you once again for being flexible with me. I know it's a little bit different from our normal, but it brings back memories. So uh, I'll see you all on Wednesday. Hopefully you all stay safe out there and don't get your internet disconnected and everything. But uh, yeah, I think everything should be back to normal by then. Alrighty, so let me stop the recording.